Hey guys, welcome to mini lecture number 3 on muscles. So what we're going to start with today is we're going to take an in-depth look at a muscle cell, in other words, a muscle fiber. Right here on the screen, we see a single solitary muscle fiber, and we can see some of its components as well. Now, I want to point out a few things. First of all, notice that we have let's get an arrow there. We have 1, 2, 3 nuclei. Remember, a muscle cell or muscle fiber is a syncytium. That means it is one cell made from the fusion of several cells together. So it is a multinucleate cell. Now, what else can we see here? We can see the membrane, the plasma membrane of the skeletal muscle cell. It's been reflected back here, and we can totally see it going all the way around. That is the plasma membrane. It is called the Sarco Lemma. Sarco means flesh, lemma means covering. Remember, we had an axolemma a while back and a neurolemma. So the plasma membrane of the muscle cell is the sarcolemma. Now, everything inside that muscle cell is going to be the sarcoplasm. Think about that as being the equivalent of cytoplasm, but just for a muscle cell. All right, inside this muscle cell, inside the sarcoplasm, there are 4 things that I want you to know about right now. We'll add some more stuff later, but 4 things right now. 2 of them we can see. We can see myofibrils. Myofibrils are these rods of protein. They are actually the contractile machinery. So, we already know 2 of the major proteins that are going to make up myofibrils. 2 of the major proteins that are going to make up myofibrils are actin and myosin. There are other proteins that we'll meet later. So these 2 proteins, which we called contractile proteins, are in the myofibrils. So the myofibrils are going to function for contraction. They are these cylinders of protein, and I see so many in here, too many that I don't want to bother counting them all. I want to point out one more thing about the myofibrils. Check out that there is a repeating pattern here, right? We actually see the striations in the myofibrils. If we look over here, for example, we can definitely see the striations. Dark, light, dark, light, dark, light, etc. Okay, what else can we see? We can see mitochondria. A lot of mitochondria. This totally makes sense because mitochondria make ATP, the cell's source of energy. Muscle cells, of course, will need to burn a lot of ATP to use a lot of energy to do the work that they do. All right, couple other things that are in here, but we don't see them. There is a protein called myoglobin. It is an oxygen storage protein. So your muscle cells actually store oxygen. The muscles don't always want the respiratory system to have to catch up. The muscle cells sometimes want to go, 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 and they need oxygen at the ready. So myoglobin will let them store oxygen. Now, that's not the only thing we're storing. We are also storing glucose. Glycogen is a branched polymer of glucose. So it's all these glucoses, and it has branches to it. Dun, dun, dun. And the branches will have branches of their own. The more branched you are, the more surface area you have, the easier it is to chop off a glucose. Well, we know glucose is a sugar that we can use to make ATP, and we know our muscle cells need ATP for energy. All right, okay. Let's go on to the next slide. Here I'm looking at some skeletal muscle cells, muscle fibers, under the microscope, and I can see those dark and light bands. I got a dark band, and I got a light band with a line in it, dark band, light band with a line in it, dark band, light band. Those are my striations. And my striations are the result of the repeating pattern of the arrangement of actin and myosin. And actually name the visible bands that I see. The lighter one is called the I band. Notice that the light has an I band. That's not where the name comes from, but it's a good way to remember it. The darker one is called the A band. And we'll learn more about this soon enough. Now, one thing we can do is we can divide the muscle fiber into these structures called sarcomeres. The sarcomere it's called the contractile unit. Every muscle fiber will have millions of sarcomeres. Each sarcomere is a place of contraction. So if we can figure out how a sarcomere contracts, then we'll know how the muscle cell contracts as a whole. And we're going to go into detail on these guys. So I'm just kind of making introductions. It's just the, how are you? Pleased to meet you part that we're doing right now. All right. What I'm going to do now is start to look a little bit more at the myofibril. So the myofibril, we see it up here. This is, let me put an arrow on it, this is one myofibril. Every muscle fiber has lots of myofibrils. The myofibrils have actin and myosin as two of their main components. Now, that one myofibril that I just labeled, it's blown up down here. This is another myofibril. And remember, the myofibril had striations. We can see the striations on it. So we can see the striations here as well. And the striations are the result of the arrangement of actin and myosin. Okay, in this diagram of myofibril, the blue lines are made of actin. All right, it says the thin filament, actin. So the blue lines, they're these things called thin filaments. These horizontal blue lines here are thin filaments, and they're made of actin. The thicker, redder horizontal lines, they're known as the thick filament, and they're made of myosin. Okay, check this out. Right here we have a region of just thin actin filaments, right here. Thin actin filaments. Then we have a region where we have the thick filament. And in the part where we have the thick filament, there is a region of overlap, then just thick, then a region of overlap. Then we have another region of thin. And then it repeats itself, and there's this repeating pattern. Now, the part that just has the thin actin filament right here, this part right here, is your I band, the lighter part under the microscope. The part that has the myosin and had a region of overlap, a region of just myosin, and another region of overlap, that trio right there is called the A band. So check this out. I band, A band, I band, A band. Now, these actin and myosins need to be anchored. And they are. There is a network of vertical proteins called the Z disc that anchor my thin actin filaments. This zigzag here is the Z disc. Doesn't stand for zigzag, it stands for Zwischenscheibe, but we don't need to know that. So the Z disc stabilizes the actins. The myosins are stabilized by this triad of proteins, this trifecta, this trio, called the M line. And this region where we have just myosin is called the H